When you think about the body's healing process, you usually picture a highly organized, efficient system.
Right, like a well-oiled machine.
Yeah, exactly. You get a cut, the immune system rushes in, builds a little scaffold, patches it up, and then it goes home. It's precise.
I mean, it's supposed to be a temporary, highly regulated emergency response. The paramedics arrive, do their triage, and clear out so normal function can resume.
But imagine if, instead of just patching a pothole, that emergency crew showed up and completely paved over a quiet neighborhood. Like they built a massive, permanent 10-lane superhighway.
Oh, wow. Yeah.
And worse, imagine they hook that superhighway directly up to your pain receptors.
That is a terrifying biological scenario.
It really is. But it isn't science fiction. It is the exact physical reality for women suffering from provoked vulvodynia. And today, for this deep dive, we are figuring out how the immune system gets tricked into building it.
Specifically, we're looking at how elevated pro-inflammatory cytokines in the vaginal microenvironment dictate the physical density of local pain-sensing nerve fibers.
Right. And for you listening, provoked vulvodynia, or PV, is a chronic pain disorder characterized by severe localized pain in the vulvar vestibule. And it's provoked by normally non-painful things.
Yeah, light touch, pressure, just friction from clothing.
And for decades, the physical cause was super elusive. People were often incorrectly told it was psychosomatic, like it was all in their head.
Which is so frustrating because when you look at the histology, the tissue samples, you see an undeniable, profound structural rewiring of the local tissue.
It's physical. The hardware actually changes.
Absolutely. We are talking about a localized immune response that just goes entirely rogue. It abandons defense and pivots to unwarranted nerve growth.
Well, let's get right into the physical scale of this, because the sources we have, the histology you're mentioning, it's staggering.
It really is.
When researchers take vestibular biopsies from women with PV and run computerized histomorphometry, specifically using an S100 protein stain to, quantify the nerve tissue.
Right. The S100 stain highlights the nerves beautifully.
Yeah, so they aren't just finding slightly irritated nerves.
Yeah.
They are finding that the density of these nociceptive fibers, these pain nerves, is up to 10 times greater than in healthy controls.
10 times the physical hardware. I mean, the S100, along with PGP 9.5 stains, it removes any diagnostic ambiguity.
The proof is right there under the microscope.
Exactly. You are looking at a localized region where the biological blueprint has been radically altered. But what is truly fascinating and a bit unsettling is the morphology of these nerves.
What do you mean by morphology? Like how they look?
Yeah, their shape and structure. This
Yeah.
isn't the orderly, linear nerve growth you see during normal fetal development. The biopsies reveal extensive, what they call collateral sprouting.
Okay, let's unpack that.
Yeah.
Because collateral sprouting usually implies severe trauma, right? It's chaotic, frantic branching.
Right.
You usually only see that kind of disorganized nerve growth after a major physical severance, like an amputation, where the severed nerve ending is desperately trying to reestablish a connection.
Precisely. It strongly resembles a neuroma. But here, we have an intact mucosal environment. There is no severed limb. There's no massive physical trauma.
So why is the body building these extra roads in the first place? What triggers this massive construction project if there's no injury?
The answer lies in the microenvironment itself, which in these cases is absolutely saturated with pro-inflammatory cytokines.
Let's break down that saturation for everyone, because it's not a general systemic inflammation like you'd get with the flu.
Yeah.
It's an intense, highly localized chemical storm.
Right, localized to the vulvar tissue.
When there's recurrent inflammation in that specific vaginal microenvironment, say, from repeated yeast infections or other chronic irritants, you get a massive influx of immune cells.
Specifically, mast cells and macrophages. They just flood into the subepithelial stroma.
And when those mast cells and macrophages arrive, they degranulate. They release a highly potent, localized chemical payload.
A chemical soup, really. We are looking at elevated levels of interleukin-6, interleukin-1 beta, tumor necrosis factor alpha.
The classic inflammatory heavy hitters.
Exactly. These are the classic drivers of the inflammatory cascade. But the critical mediator here, the one that bridges the gap between immunology and neurology, is nerve growth factor, or NGF.
Wait, nerve growth factor. So if they are actively dumping nerve growth factor into the tissue, the immune system is essentially acting as biological fertilizer.
That's a great way to put it, yes.
It's not just incidental damage from the inflammation. It is a specific chemical instruction for those local pain nerves to branch out and multiply.
It's a direct command. NGF binds specifically to the chi-Ka receptors located on the terminals of these nociceptive neurons.
Okay.
Now, under normal, healthy conditions, a tiny amount of NGF is required just for the maintenance and survival of sensory neurons. They need a little bit just to live.
But in the context of PV,
In PV, the mast cells and macrophages are continuously pumping massive quantities of NGF into the extracellular space.
wow. And if I remember the intracellular mechanics correctly from the sources, when that flood of NGF binds to the receptor, it gets internalized into a signaling endosome, right?
Yes, that's right.
And that endosome travels all the way back up the axon to the neuron cell body to actually alter gene expression.
That retrograde transport is exactly what happens. It hits the nucleus and triggers the upregulation of structural proteins. The neuron starts producing the building blocks needed to physically build new axonal branches.
Hence the collateral sprouting and the tenfold increase in density.
Exactly. But the complexity of this microenvironment doesn't stop at mere growth.
Right, because to cause provoke surface pain, these nerves have to be where the stimulus is.
Right. And normally the extracellular matrix acts as a physical barrier. It sequesters nerves to the deeper stromal layers. They shouldn't be sitting right on the surface.
So how are these newly sprouted branches breaching that barrier? How are they getting to the surface?
They are using enzymatic drills.
Enzymatic drills.
Yeah, mast cells in this inflamed microenvironment release an enzyme called heparinase, or HPSE.
I remember this from the paper, which degrades heparin sulfate proteoglycans in the extracellular matrix.
Yes. HPSE cleaves those proteoglycans, literally breaking down the structural integrity of the tissue barrier.
It just chews right through the scaffolding.
It does. And this allows the newly formed, highly sensitive nerve fibers to penetrate right up into the superficial epithelium, the very outermost layer of the skin there.
Where they absolutely do not belong.
Right. Furthermore, heparin sulfate normally binds and sequesters growth factors, keeping them inactive.
Oh, wow. So when HPSE cleaves it,
It unleashes even more bound growth factors into the tissue, creating a vicious, localized feedback loop of continuous growth and structural degradation.
okay, so let's summarize where we are. We have 10 times the normal amount of pain nerves. They are aggressively sprouting, and they are enzymatically drilling their way to the absolute surface of the tissue where they are completely exposed.
That's the anatomical reality, yeah.
But here's a variable that doesn't quite make sense to me. Millions of people experience recurrent localized inflammation.
Sure, yeast infections are incredibly common.
Exactly. But millions of people do not develop a permanent 10-lane superhighway of pain nerves. There has to be a catalyst that dictates who gets trapped in this feedback loop, right?
There is, and the divergence lies in the genetics. If we look at the genetic profiles of women with PV compared to healthy controls, there's a huge difference.
What are they finding?
There is a significantly elevated prevalence of specific single nucleotide polymorphisms, or SNPs, in the genes that code for NGF, and crucially for a pain receptor called TRPV1.
So it's a genetic predisposition to an exaggerated neuroimmune dialogue.
Exactly.
If you have these specific genetic markers, your baseline regulatory mechanisms are already altered. Your immune cells are literally genetically programmed to overproduce nerve growth factor the moment they encounter an irritant.
You nailed it. The threshold for initiating this wild nerve growth is inherently lower. A minor inflammatory event that would normally resolve quickly, like in a few days instead, triggers a disproportionate overreaction.
The tissue floods with NGF.
Right, which not only causes the nerves to sprout, but actually acts as a chemoattractant for more mast cells, which in turn release more NGF.
A total runaway train.
Yeah.
Which brings us to the actual mechanics of the pain itself.
Yeah.
Because having more nerves is really only half the equation, right?
Right, it's not just about nerve density.
The sources show that the inflammatory soup doesn't just build the hardware, it fundamentally alters the software. It changes how these nerves behave.
This is where we get into allodynia.
And for you listening, allodynia is where a completely innocuous stimulus, like the friction of clothing or just the light touch of a cotton swab, is perceived by the brain as severe burning pain.
The mechanism of allodynia here is driven by the upregulation of specific pain channels, primarily TRPV1 and TRPA1, on the membranes of these newly sprouted nerve endings.
Here's where it gets really interesting. It's not just that there are more alarm bells because of the extra nerves. It's like the immune system has rewired the bells so that a gentle breeze sets them off instead of a heavy hammer.
Rewiring the bells is a great way to think of it. They're installing hair triggers.
Hair triggers, I like that.
Yeah, because TRPV1 is normally a high-threshold receptor. It's the exact same channel that detects extreme heat, like a chili pepper, or a physical burn.
Right, it takes a lot of energy to open it.
Exactly. It requires a noxious level of thermal or mechanical energy to open the ion channel and fire a pain
Medical.
signal.
But when you bathe that nerve ending in NGF, interleukins, and histamine from the immune cells,
That threshold plummets dramatically. The intracellular signaling cascade triggered by that inflammatory soup phosphorylates the TRPV1 channels.
and phosphorylation does what exactly in this context?
It does two things. It rapidly traffics more of these channels to the cell membrane, so there are just more of them, and it props the existing channels into a hyper-excitable conformation.
So they are basically stuck half open.
Exactly. The threshold drops so low that normal, everyday mechanical pressure, which possesses nowhere near the energy normally required to open a TRPV1 channel, causes the channel to fly open.
The nerve fiber floods with sodium and calcium ions, depolarizes, and fires a massive, unearned pain signal to the brain.
That's allogenea at the cellular level.
So the peripheral hardware is completely hacked. You have a tenfold increase in nerve density, positioned directly at the surface with their receptor thresholds dropped to near zero.
It's the perfect storm for chronic pain.
But the downstream effects of this are where it gets truly systemic.
Yeah.
Because a 10-lane superhighway firing constant high-volume action potentials doesn't just stay local. Those signals travel up the lumbosacral spinal cord straight to the dorsal root ganglia.
And the dorsal root ganglia, or DRG, cannot act as a passive relay station when subjected to that volume of nociceptive traffic.
It has to adapt.
Right. This is the transition from peripheral neuropathy, a local tissue problem, to central sensitization. The continuous bombardment of pain signals physically alters the spinal cord itself.
The relay station adapts to the incoming volume by fundamentally changing its own architecture.
Yeah.
We're talking about robust, long-term changes in gene expression right there in the spinal cord.
Yeah, if you look at the animal models mirroring this condition, the persistent peripheral firing induces severe neuroplastic and neuroinflammatory changes centrally.
The spinal cord neurons actually begin overexpressing their own voltage-gated sodium channels and glutamate receptors, right?
Yes. And the microglia in the spinal cord activate, producing endogenous NGF and pro-inflammatory cytokines right there in the central nervous system.
So the central nervous system is basically mirroring the toxic microenvironment that started in the peripheral tissue down in the vulva.
It's rewriting its code to accommodate a baseline of continuous pain.
Which means, theoretically, if you were to somehow surgically remove all the hyperinnervated tissue in the vestibule, it might not even cure the pain.
Right, because the central servers are already hacked. The spinal cord is now primed to amplify any signal it receives or even fire spontaneously without any input.
Wow. It shifts the entire paradigm of treatment.
Completely. You can no longer just treat a localized tissue issue. You are dealing with a hyperexcitable central nervous system. This is why localized therapies often fail once central sensitization is locked in.
However, the research into how to break this specific neuroimmune cycle before it reaches that point has yielded some incredibly elegant and honestly kind of paradoxical data.
Oh, the intervention studies are fascinating.
Yeah, let's get into those. Researchers wanted to know if they could disrupt the construction crew during the acute phase. Like, if elevated cytokines are the catalyst, what happens if we pharmacologically block the immune signaling?
They utilized two highly targeted approaches during the acute phase of inflammation in these animal models.
Okay, what were they?
First, they used ketotifen fumarate, which is a mast cell stabilizer. It prevents the mast cells from degranulating and releasing that cytokine payload.
So it stops the drop of the fertilizer.
Exactly. And second, they utilized Ro8-2750, a molecule that directly inhibits NGF from binding to the TRKA receptor on the nerve.
And by deploying those during the acute inflammatory window, they effectively cut the supply chain. And the results were immediate.
Right. Very immediate. The levels of pro-inflammatory cytokines in the tissue plummeted, and the TRPV1 upregulation was completely halted.
But the structural data was the most profound part.
Right. By stabilizing the mast cells or directly blocking the NGF, they didn't just reduce the pain signaling, they entirely prevented the hyperinnervation.
The S100 stain showed the nerve density remained completely normal. The collateral sprouting never occurred.
The extra roads were never built, which meant the dorsal root ganglia never received the bombardment of signals, and central sensitization was prevented.
Okay, wait. Let me piece this together for a second. So what does this all mean? If stabilizing mast cells stops the physical growth of the nerves, then knocking out the macrophages should theoretically do the exact same thing, right?
That's the logical leap, yeah.
Yeah. Because both are heavy lifters of the immune response, both are swarming the microenvironment, and both are releasing cytokines. If we just block any immune cell, does the extra nerve growth disappear?
Well, you would think so, and that is exactly what another group of researchers tested to find out.
What did they do?
They used claudinate liposomes, which is a highly effective macrophage depleting agent, to entirely wipe out the macrophages in the local microenvironment during the inflammatory response.
Okay, so the macrophages are completely gone. Did it stop the pain?
Yes, it did. The hypersensitivity vanished and the pain responses normalized. And to quantify this objectively, they measured a marker called PERK in the spinal cord.
PERK,
Yep.
phosphorylated ERK. That's the intracellular marker that only appears in dorsal horn neurons when they are actively processing a painful stimulus, right?
Precisely. It's a direct, measurable proxy for pain hitting the central nervous system. Following the clodronate treatment and the depletion of the macrophages, spinal PERK levels dropped back to baseline.
So the central pain signaling was completely silenced.
Completely silent. But here is the brilliant contradiction, the biological paradox that changes how we view this entire ecosystem.
I'm ready. What happened?
The clodronate treatment did absolute nothing to prevent the vaginal hyperinnervation.
Wait. I have to pause you there because structurally, that makes no sense.
No, it's wild.
The pain signaling is silenced, the spinal cord is quiet, but when they look at the histology, the 10-lane superhighway of extra nerve fibers is still sitting there in the tissue.
Still there. The S100 stain showed the density of the nociceptive nerves remained heavily elevated, exactly as it was in the untreated models.
How is that even possible? If the physical hardware of the 10 times normal density of exposed pain nerves is still wired directly into the surface epithelium, why aren't the alarms ringing?
This is the critical breakthrough in understanding the extreme specificity of the immune nerve dialogue. It proves that the immune system is not a monolith.
Different cells are doing different things.
Exactly. Different cells are executing vastly different, highly specialized roles in this mythology. The data strongly suggests that mast cells, via their massive release of NGF and heparinase, are the primary architects of the physical growth.
They are laying down the asphalt and building the roads.
Right. They build the anatomy.
Ah, I see the division of labor now. The mast cells build the structural anatomy. But the macrophages are the ones maintaining the hair triggers.
You've got it. The macrophages are responsible for producing these specific inflammatory mediators that continuously phosphorylate the TRPV1 channels.
They are chemically forcing the nerves into that hyperexcitable, low-threshold state.
Right. So if you remove the macrophages with clodronate, the massive network of extra nerves is still physically present, but they are no longer being bathed in the specific chemicals required to keep them sensitized.
The channels close, the threshold goes back up, the nerves just fall silent.
You basically have a massive 10-lane superhighway, but there's absolutely no traffic allowed on it.
It's a completely dormant network. That is incredible. And this entirely reframes the pharmacological approach to localized chronic pain, doesn't it?
It does. It shows that a blanket anti-inflammatory is too blunt an instrument. If you are dealing with a patient in the early stages of PV and you want to prevent the structural rewiring, you have to target the mast cells and block the NGF pathways.
Let's stop the builders.
Exactly. But if a patient has suffered from PV for years, the anatomical changes have already occurred. The hyperinnervation is a physical reality.
So in that case, trying to stop nerve growth might be entirely the wrong target. The roads are already built. You have to focus on silencing the firing of the nerves that already exist.
Which means specifically targeting the macrophage signaling pathways to raise the TRPV1 threshold back to normal.
It is a masterclass in treating the specific mechanism of action rather than just the general symptom. It is an intricate, choreographed chemical conversation between the immune system and the nervous system.
It really is.
So to summarize this incredible journey for you listening, acute inflammation in the vaginal microenvironment doesn't just pass. It recruits mast cells and macrophages that pump out pro-inflammatory cytokines and NGF.
Right.
And this chemical soup physically forces pain nerves to sprout, multiply by up to 10 times, and enzymatically drill to the surface.
And then it makes them hypersensitive to normal touch, eventually rewiring the spinal cord itself.
It's mind-blowing. And yet, as highly specific as this immune nerve dialogue is, there is another massive biological variable at play here that we haven't even touched on yet.
Right, because this microenvironment doesn't exist in a vacuum. It is heavily influenced by endocrinology.
Hormones. This brings us to a fascinating and frankly provocative final thought to mull over. drawing on a specific detail from the texts regarding estrogen.
Yes, the estrogen aspect adds an entirely new dimension of complexity to everything we just discussed about nerve density.
The sources note that a lack of estrogen, such as what occurs naturally during menopause, or even with certain hormonal contraceptives, is strongly associated with an increased density of sensory nerve fibers in the vulva.
Which is already a huge factor. And conversely, estrogen itself is known to actively lower pain thresholds while simultaneously acting as a potent mediator of peripheral nerve remodeling.
So estrogen isn't just an innocent bystander here. It is a highly active project manager that is constantly directing how these nerves grow, retract, and respond to stimuli.
It is continuously modulating the tissue. Now, synthesize that with the mechanisms we just unpacked.
Okay.
We have established that pro-inflammatory cytokines, driven by mast cells and macrophages, are capable of physically building these dense, hypersensitive pain networks.
Right. The immune system builds the roads.
But we also know that a woman's natural hormonal life cycle involves constant, profound fluctuations in estrogen. The baseline is never static. It shifts across the monthly cycle, it shifts drastically during pregnancy, and it fundamentally alters during perimenopause and menopause.
So if inflammatory cytokines build these pain networks, and natural estrogen fluctuations also constantly remap nerve density, how might the natural hormonal cycles across a woman's lifetime be secretly interacting with her immune system to completely redraw her body's architecture of pain?
That is the frontier of this research. Are the immune system's structural changes being exacerbated by the cyclical drops in estrogen?
Like, are these two highly powerful biological systems, the immune response and the endocrine system, fighting each other for control of a local architecture? Or are they colluding to lock the tissue into a permanent state of hypervigilance?
It scales everything we just learned about a microscopic cellular interaction up to the timeline of an entire human life. It suggests that provoked vulvodynia and perhaps many forms of localized chronic pain aren't just the result of a static historical injury.
It is a dynamic, ongoing daily conflict between entrenched immune memory and inevitable endocrine shifts.
The structural hardware might be built by cytokines, but the operational software is being constantly patched and rewritten by hormones. Untangling that specific web is the next crucial step in identifying a true restorative cure rather than just symptom management.
Which means there are still so many incredible layers of this cellular machinery left to uncover. The fact that the body can physically rebuild its own neural networks in response to microscopic chemical cues is just, well, it redefines how we have to think about pain.
Absolutely. It's not just a signal, it's a physical structure.
Keep exploring the mechanisms, keep questioning the baseline assumptions, and never underestimate the immense adaptive complexity of human biology. Thank you so much for joining us on this deep dive. We will catch you on the next one.